All right, welcome everybody. As you guys can see by the tabs at the top of my screen, we're on week 13, which means we only have this week and next week, and then we're completely done with the weekly reviews and honestly with the content for Biochem 1. So congratulations to you all for making it this far. So before we jump into the review itself, I want to just briefly mention what's coming next. So of course, we're going to be talking about the tricarboxylic acid cycle, citric acid cycle, Krebs cycle, whatever you want to call it. There's about a million other names for it. Today, we're also going to talk a little bit about some of the alternative pathways and things like plants and stuff. And then next week is all about the electron transport chain. And next week, I think, is one of the more complex weeks. It's very, there's a lot of details that aren't quite fun to memorize. So next week is going to be a little bit challenging. And then, I mean, we're done with reviews because that's also the last week of classes, and then you have finals the following week. Okay, so jumping into the review, I want to briefly mention I'm going to kind of stray from my usual format. Normally I make questions and we talk about them, but for this kind of content, I think that it's too difficult to make questions that encompass everything. So I'm usually just going to be going over the PowerPoints just very briefly. I do make slight modifications, I do add my own notes, and we will be talking about things in kind of, I'll be emphasizing the content that I would encourage you to focus on. But yeah, so that's why rather than questions like I normally do, you're seeing what appears to be the first slide on some of the professor's notes. So yeah, so once again, like I mentioned, I will emphasize the content that I feel needs to be emphasized. I will do my best to kind of make that clear as well. I might try to add so this is a kind of question you could be asked every so often. But yeah, so let's go ahead and jump in. So we've been talking so much about glucose and pyruvate. Now we finally get to do something with the pyruvate. So the key here is, like we mentioned, so anaerobic glycolysis has that 11th step. In humans, it goes to excuse me. Humans, it goes to lactate. In I believe yeast and bacteria and stuff, it goes to alcohol. Excuse me. And so in the absence of oxygen, pyruvate is reduced to lactate, NADH is reoxidized to NAD+ for further use in glycolysis. Okay. In the presence of oxygen, it's a little bit different. When you have oxygen, pyruvate gets oxidized completely to carbon dioxide. So that's something that I'm going to be pointing out a lot when we go through the actual Tca cycle, is how each carbon becomes carbon dioxide, because it's not all at once. Okay. So reducing equivalence. So NADH, NADPH, which we talked about last week with the pentose phosphate pathway, but specifically NADH and FADH2 are made and then reoxidized by the electron transport chain, right? So this is called oxidative phosphorylation. And I want to draw this to your attention, partly because it is one of the bold words that you need to know, but also because up until now, everything we've been doing has been substrate level phosphorylation. So when we I guess I'll call it the break-even point of glycolysis reaction, was it 7, I believe? That was a substrate level phosphorylation because we had some carbon structure, some organic structure, phosphorylate ADP into ATP. Now, oxidative phosphorylation in the electron transport chain, which we're covering next week, is going to be H2O to O2. Okay, so oxygen is the final product of the electrons. That's what makes it oxidative phosphorylation. Okay. And then, as you see there, respiration is the metabolic process of ATP production. And so, yeah, okay. Oh, I apologize. I have this backwards. It's oxygen goes to H2. My apologies. That way, excuse me. Okay. So we have covered up until this point very well. Glucose to pyruvate, we know backwards for gluconeogenesis. We know everything we can possibly know about it. This is where things get complicated. So we're very briefly going to go over Pyruvate Dehydrogenase. That's the enzyme that catalyzes the dehydrogenation. And then we're going to go over the Krebs cycle, or the Tca cycle. Next week is the electron transport chain, as I hope you guys can see from this figure, it is intensive. Okay, so I'm going to be zooming in for this part because we're going to actually just go straight into the Tca cycle now. So prior to entering into the Tca cycle, we make acetyl Coa. Okay. In eukaryotes, this all happens in the mitochondria. This is really important. I cannot emphasize this enough. We talked about compartmentalization in the chapter on metabolism. The fact that it happens in the mitochondria is crucial. Okay. All right. Anyways, so we start with our Acetyl Coa. So one thing that's important to remember is pyruvate has 3 carbons, and I'm going to be using C to represent number of carbons. Acetyl Coa brings us down to 2 carbons. Okay? Why? Because the first carbon is completely oxidized into carbon dioxide. Okay? All right. So we talked about oxaloacetate in the gluconeogenesis pathway. And one of the big things we talked about with oxaloacetate is the enzyme to make it is already present, right? Or the enzyme for malate especially is already present, right? But let's talk a little bit more about why oxaloacetate is present. Oxaloacetate, so think of the entire pathway, or I should say the reason the Krebs cycle is a pathway is because we start with 2 carbons here and 4 carbons in the pathway. So we're introducing 2 more carbons into this cycle, and then we bring it down to 4, and then we get another 2, and we bring it down to 4, and we get another 2, and that's how the cycle works. So oxaloacetate is kind of the first reactant in the cycle that we use. Citrate is the first product in the cycle, okay? And we're going to go all the way back until we get back to oxaloacetate, okay? So a couple of keys here is and we're going to talk more about the reactions in depth. This is a fairly simple reaction for citrate synthase. What we're doing is we are basically, how do I word this? We're undoing a hydrolysis, if that makes sense. We're undoing the condensation reaction. So by introducing water, we can bring this carbon, I was trying to think how to represent it, but this carbon eventually bonds to, if I'm not mistaken, it is this carbon. Yeah, it's this carbon. Sorry, it takes me a moment to recognize that. Okay, and we kick off the CoA. So CoA is coenzyme A. Enzyme A. It's a big organic mess of a structure. You don't need to know that much about it, just that it exists, just that it's used. Okay? All right. And it's going to come up again in the future, just so you're all aware. All right? And the SH is this sulfur. So it's SH, so it's a thiol group. It just makes it a leaving group. It's not that big a deal. Okay, yeah. All right. So now citrate to isocitrate. This is a fascinating reaction because at the end of the day, it's just an isomerization. Okay? No, we're going to talk about that in the reaction. I love this reaction because I think it's brilliant. It doesn't make sense why the enzyme is called this until we talk about it in more depth. But I'm going to get there when I get there. I don't want to get ahead of myself and talk about some reactions, not others. We'll talk about it later. Okay. So this is the first reaction of the Tca cycle where we actually use carbon dioxide, or where we actually produce carbon dioxide, excuse me. Okay. So carbon dioxide is here. And I also want to draw your attention to the name. Isocitrate dehydrogenase. Obviously, it's a very easy name, but also when we made isocitrate up here, oh, excuse me, I accidentally scrolled to the top. Here we go. When we made when we made carbon dioxide up here, the enzyme we used was pyruvate dehydrogenase. Okay, I hope you guys are seeing a trend. They're all dehydrogenases, or let me reword that. We tend to have dehydrogenases when we have carbon dioxides. There's one exception in the cycle. That I'm just pointing out right here, that's malate dehydrogenase. But for the most part, keep doing that, my apologies. For the most part, CO2, dehydrogenase, okay? Anyways, right, so one thing that I've been forgetting to keep track of, so I apologize for that, is we had 4 carbons and 2 carbons from oxaloacetate and acetyl CoA. Citrate is 6 carbons. We just added the 2 together. Isocitrate, like I mentioned, it's an isomerization reaction. We're not gaining or losing any carbons because it's just moving stuff around. It's also 6 carbons, right? We've lost 1 carbon here as CO2, so alpha-ketoglutarate has to be 5 carbons, right? And this is alpha-ketoglutarate, by the way, right? The alpha-ketoglutarate dehydrogenase loses us another CO2 group, so the Succinyl Coa is 4 carbons, okay? All right, now speaking of Succinyl Coa, this is really interesting. The CoA group, this isn't the same one as above because this one is both like lost and gained by the exact same substituent every time. But this CoA group binds to, or I should say, it takes the place of the CO2 effectively, right? So we kick off this group right here, and in that same position, we have the S-CoA from acetyl CoA. Or I'm sorry, not acetyl CoA, I keep doing that. The coenzyme A. I apologize. I've been saying acetyl CoA this entire time for succinyl CoA. That's incorrect. The coenzyme A. I apologize. So in the place of this CO2 is coenzyme A. Okay? And then it gets kicked off again in this succinyl CoA synthetase. Okay? So this is one of the first reactions that I think is really weird. And it's really weird for kind of 2, maybe 3 reasons. The first is the enzyme name. We use synthetases to synthesize, and yet this synthetase is taking the thing that it should synthesize and making it something else, okay? So we start with Succinyl Coa, when Succinyl Coa synthetase intuitively would synthesize Succinyl Coa, not break it down. Okay, that's something that I think is rather interesting. It's also a little bit weird. And the answer to this question is the exact answer that we talked about with glycolysis and gluconeogenesis. This reaction is, I mean, you can use energy, you can couple it with energy, or you can uncouple it from energy, and you can get either the forward or the reverse reaction, okay? So because this reaction can go in either the forward or the reverse direction, depending on the energy, the synthetase is still there, it's just acting in reverse. Okay, so that's the first weird thing that I think about, or that I think this reaction has. The second is we talk about GTP and GDP, and this whole mess, right? It doesn't. So here's the deal. I told you all in the, let me think. I don't remember when I mentioned it, but in one of the units when we were talking about other triphosphate nucleotide bases, so ATP, GTP, UTP, and CTP. I mentioned those very briefly, and I said, let me give myself a little bit of space at the bottom, excuse me, okay. And I said, XTP has a delta G naught of the hydrolysis in pretty much every case of about negative 30.5 kilojoules per mole. And this concept right here is why that's the case. Okay, oh, I apologize, okay. Because, and I'm gonna erase this, I apologize, I used the blue for the confusing parts, but it's just too much going on. Okay, so, because this reaction couples GTP synthesis to Succinyl Coa breakdown, we get GTP out of it, okay? It's a what's it called? It's a specific reaction that it requires specifically guanine, or really it's guanidine nucleotide in order to work. So since it's requiring that guanine, then that can be used to make ATP. Okay, because they have about the same energy, if we couple the hydrolysis of GTP to the synthesis of ATP, it ends up being about equal, and we can get everything in terms of ATP, right? That way we can kind of compare things much more equally, or much more easily, excuse me. So this is called Nucleoside Diphosphate Kinase, and let's think about the name once again. Let's break it down. Nucleoside refers to adenosine, guanidine, guanidinosine, excuse me. I don't like the guanine's version of adenosine. But the S, rather than nucleotide bases, it's a nucleoside base, is because just like we change adenine to adenosine is the same thing. Diphosphate, because you start with an ADP kinase, you add the phosphate to that diphosphate. Okay, and that's where you get the triphosphate. So what this reaction is basically telling us is the synthesis of succinate from Succinyl Coa releases enough energy to make GTP, which can then be used to make an equivalent number of ATP from it in a reaction catalyzed by nucleoside diphosphate kinase. Okay, that's it. I know it's a little bit complicated. I know that it's not the most intuitive, but that's it, right? As to where the phosphate's coming from, it starts as an inorganic phosphate, okay? All right, so let's move on. So we've messed around with phosphates. There was energy, there was coenzyme A, but what we did not do is change the carbon number. So succinate makes sense, it has 4 carbons. All right, so now succinate dehydrogenase is another one of the exceptions, and I will highlight it rather than yellow, since I've been using that to show the combination of dehydrogenase and CO2, we will use orange, I guess. Okay, so dehydrogenase does not look as good as I'd hoped it would. My apologies, everyone. I guess purple maybe? Oh god, that looks even worse. We are just going to stick to green. Green will do for these. So this is a dehydrogenase that does not release CO2. Okay? Now I think that it can be a little bit weird that it doesn't release CO2, but the reason that it doesn't is because it doesn't have to. And I know that's not the most intuitive, but here's the thing. I told you guys in this area right here, for each I shouldn't say for each area, but for each of these, we have two carbon units, okay? We need to bring down the 2 carbon, we need to release the 2 carbons. So we only needed to release 3 carbons from pyruvate. I'll make a running tally, I guess. So this is carbon number 1 of pyruvate. This is carbon number 2 of pyruvate, or number 1 of acetyl CoA, and this is carbon number 3 of pyruvate, or again, number 1 of acetyl or number 2 of acetyl CoA. Therefore, we don't need to release any more carbons, and that's why we're not going to. Okay? All right. So succinate dehydrogenase, this also is kind of a weird reaction for a different reason. Whereas, and I've kind of glossed over this, but in most of our dehydrogenase reactions, we're getting NADH. We get NADH here, we get NADH here. These are both of our dehydrogenases, and we also got one up here, NADH. Okay, this one, because we're not releasing the energy from the carbon, gives us FADH2. And FADH2 has less energy. Okay, all right. So now, next reaction is fumarate to malate using fumarase. Now, I think this kind of makes sense because they sound the same. Fumarate, fumarase. I mean, they come from the same word. My apologies. I meant to do Control-Z. There we go. And it uses water. Okay. Now, this is the last dehydrogenase and also the last reaction of the Tca cycle. And this is malate dehydrogenase, converting malate back to oxaloacetate. And we get NADH from this. Now, this does not release CO2. Oh, hang on, I just crossed out no CO2. Yes, there is no CO2. Yeah, there we go. All right. Now, the reason that this can produce NADH, even though it's not produced, or hang on, sorry, let me reword that. The reason this can produce NADH despite not releasing CO2 like the others, like for example, the isocitrate dehydrogenase reaction, CO2 and NADH go together. The Alpha-Ketoglutarate, same thing. The Pyruvate Dehydrogenase, same thing. This is the exception. This is why it's also a weird reaction. Okay, it comes down from what we'll talk about that later. We'll talk about the details later. Actually, I don't want to get too deep into the weeds. Okay. Last thing is this is also 4 carbons. Okay. So we're talking about this just as a brief overview. We are obviously going to go into a lot more depth about it, but very briefly, I just want to bring your attention to this piece on the bottom left. Each turn of the cycle liberates 2 CO2, the 2 in acetyl CoA, 3 NADH, 1 FADH2, and 1 ATP. And we'll be talking about NADH and FADH2 in more depth next week because that's when we'll be talking about taking those those reduced products and reoxidizing them in oxidative phosphorylation. But the reason I mention this is twofold. First, this ATP is actually a GTP, and we're treating it as ATP equivalents because they are equivalent and because it will convert to ATP because we're doing this for energy. The second thing is, once again, the key thing here is each turn of the cycle. Okay, up here we talk about two carbon units enter the Tca cycle. In other words, double this if you want it per glucose molecule. Okay, so we're going to do kind of an inventory at the very end of this PowerPoint, and we're going to say per 1 molecule of glucose, what did we get out of it? Okay, and the reason I want to do it at the end is because we very briefly talked about pyruvate dehydrogenase kind of existing, and then we moved on, and we haven't gone into depth about any of these reactions yet. Okay, so let's go ahead and jump in. So this, I don't think it's ever been asked about if I'm being honest, but if you guys wanted to know, this is a coenzyme A. Cool, doesn't really matter too much. Okay, so this is the or I guess this isn't even the first reaction. This kind of applies for the first reaction with acetyl CoA and oxaloacetate, but basically what this slide is telling you is just this sulfur makes for a good leaving group. So if you have a nucleophilic attack from a strong enough nucleophile, thinking back to OCHEM, this can take the electrons and be kicked off easily. Okay, because of that, coenzyme A is considered activating for a substrate. And when I say activating, I mean it makes it more reactive because it now has a better leaving group that can be used. And so this is more favorable than hydrolyzing an oxygen ester. So if it was, and let's see if I can draw this, CoA bound to an oxygen, bound to this whole thing, this reaction would need a very specific nucleophile. It would need a much stronger nucleophile in order to happen. This reaction is much more general. Okay, so if you guys remember chlorination versus bromination from OChem, it's a similar concept. I don't want to get too caught up in that, so I'm just going to move on, but yeah. Okay. And here are the energy values that prove that, by the way. So for effectively the exact same thing, so CH3, carbonyl, sulfur, something else, CH3, carbonyl, oxygen, something else, when you react it with water, this is spontaneous. It's quite spontaneous even, negative 20 kilojoules per mole. This is even more spontaneous. Okay, so it makes it energetically more favorable, and so it's our body being more efficient. Okay, all right. So thiamine pyrophosphate, excuse me. So I guess I should say thiamine, to start off with, is required for thiamine pyrophosphate. Thiamine pyrophosphate catalyzes the decarboxylation of an alpha keto acid. So let's break down what an alpha keto acid is. Okay, so first, the acid refers to a carboxylic acid. Okay, and then the alpha keto is kind of a modifier of the carboxylic acid. Okay, so alpha is saying one carbon away. So this is the carbon of the carboxylic acid. This is the alpha carbon to it. Keto refers to not the keto diet, as kind of pops into our heads usually, but it refers to a ketone. In other words, a keto acid is just one that has a carboxylic acid and then another carbonyl directly attached to it. So we talked about one keto acid, although we didn't name it as such. We talked about alpha-ketoglutarate. It has that name for a reason. And that reason is that, there it is that right here is the keto acid. You have the carboxylic acid group here, and then on the carbon directly attached, you have a carbonyl. Therefore, this is an alpha keto acid. Okay, oh, I apologize. I forgot that I was zoomed in. Okay, so the reason they bring this up is because we have a decarboxylation of an alpha keto acid. We have the reaction from alpha-ketoglutarate to, I hope you guys remember, but I won't blame you if you don't, since we just talked about this, alpha-ketoglutarate to Succinyl Coa. Succinyl Coa. This releases CO2, and so this is a decarboxylation of the alpha keto acid. Therefore, without learning anything else about this reaction, I know that the alpha-ketoglutarate dehydrogenase enzyme has to have something to do with thiamine pyrophosphate. I don't know. Excuse me. I don't know what, but I know it has something to do with it. Okay. So let's move on. And this is a great example of exactly what we were talking about. I'm not going to waste too much time here. Okay. All right. So this is lipoic acid. Admittedly, I don't remember exactly why this is covered. I don't think we need to worry about this too much. Oh, it's just talking about the how they can work to help in oxidation decarboxylation of alpha keto acids. Very similar concept to above. Effectively, what they do is they couple acyl group transfers. So an acyl group is basically just a carbon chain, and an electron transfer. So this H plus that you see at the end, the what is it called? The reduction of NAD plus to NADH, that's kind of catalyzed by a lipoic acid. Okay, all right. Now let's get into the nitty-gritty details we have to memorize. The stuff above is helpful, but not super necessary. Pyruvate dehydrogenase is necessary. Okay, so pyruvate dehydrogenase is a 3-enzyme complex. Okay, and we're just going to go in order, 1, 2, and 3. The first is pyruvate loses CO2 and forms excuse me, hydroxyethyl thiamine pyrophosphate. Okay, so that's what HETPP, by the way. That's what that is. Let me write that down. Hydroxyethyl, and I'm just going to say this thing because I don't want to write out thiamine pyrophosphate. Okay, so step 1, pyruvate is decarboxylated to TPP to form hydroxyethyl TPP. Makes sense. Okay, second step. The hydroxyethyl group is then transferred to lipoic acid and oxidized to form acetyl dihydrolipoate. Okay, now this is kind of wordy, but all that's happening is, well, let's think about what a hydroxyethyl group is. An ethyl group is a 2-carbon group. Hydroxy is a hydroxyl group. So this is hydroxyethyl. This is also this hydroxyethyl group, to be more specific. Okay, so it's transferred to lipoic acid, makes sense. And oxidized, how does it get oxidized? We get an extra bond to oxygen. So it turns from a hydroxyethyl to, effectively a ketone. In this case, it would be a, actually, I'm not sure exactly what it's called with the sulfur. Let's not go there. All right. Then we've talked about sulfur before. Cleaving a thioester, well, that's the exact same thing as the CoA group that we talked about. Cleaving this thioester takes a lot less energy. So acetyl CoA can come in, attack here, kick this off, and form acetyl CoA. Excuse me. So those are the 3 steps that we care about with respect to pyruvate. However, there are steps 4 and 5, as you guys can see here. And these are important for NADH. Okay, so step 4 reoxidizes. Oh, I'm sorry, not step 4 and 5. It's step 4, but it's split up. So step 4, lipoic acid is reoxidized. So it's reoxidized into a ring structure between the 2 sulfurs. And the way they do that is using FAD and FAD and NADH. So that's how we get our NADH out of pyruvate dehydrogenase. Okay, and once again, this is a reminder. This is per 1 pyruvate, we get 2 per glucose. Okay, and I think it's important to recognize this because in glycolysis, we've been doing everything in terms of units of glucose, or like 1 molecule of glucose gives us 2 ATP. It gives us 2 NADH, it gives us blank, right? This time we're doing everything in terms of pyruvate, and so we have to double everything. So I'm saying 1 NADH, but if we think about it from the perspective of glucose, it's 2 NADH, okay? And I'm not going to read all of that in detail. All right, but there is more detail that I haven't mentioned. Actually, this is just your professors going through each step. I'm not going to spend too much time on this. Okay. So yeah, so it's a large, complex, multi-subunit structure, and it's got plenty of subunits. The subunits aren't asked about too often, but we can just very briefly talk about it. We get lots of E2 subunits. So E2 is the one on the left, so you guys can see. E1 is a tetramer, and E3 is a dimer. E1 is a monomer, okay? And in my mind, it kind of makes sense that we get more subunits of E2, because since it's just a monomer, it's kind of more simple. Easier to synthesize and kind of easier to put together, okay? So that's another picture. This is it kind of all at once, okay? So there's kind of a couple important things to mention here. Is first, lipoamide is a swinging arm. What does that mean? Basically, lipoamide is kind of what you see in E2. Excuse me. It's the end of the lipoic acid. It's this part right here. It sticks out, and so as it rotates, it can reach complex 1 and it can reach complex 3. Or I'm sorry, it can reach enzyme 1 of the complex and enzyme 3 of the complex. Okay, and it's obviously really close to enzyme 2, right? And enzyme 3 is a flavo enzyme. Okay, so this means that they use FAD and FADH2. Okay, that's it. It's not a super complex meaning. It's just something to keep in mind because those do come up again. Right? And here's another picture. Admittedly, I don't like to focus on these too much. This is what I like to focus on. So here we are at the first step of the electron transport, I'm sorry, not the electron transport chain, the tricarboxylic acid cycle. Citrate synthase catalyzes a carbon-carbon condensation between acetyl CoA and oxaloacetate. The product is citrate, which is a tricarboxylic acid, hence the name tricarboxylic acid cycle. So a couple things to point out are, first, this enzyme is using a base to abstract 1 hydrogen and then to hydrogenate. And then from there, this negative charge can come in and attack and quickly attach. Fairly straightforward. Okay, and then finally the CoA is kicked off in the second step. So you get Citrate CoA as an, I'm sorry, not an intermediate, a transition state, and then you get citrate at the end. Okay, this is a very spontaneous reaction, more spontaneous than ATP hydrolysis. That makes it pretty much irreversible under our body's conditions. So when you're going to go through the tricarboxylic acid cycle, think of this as the committing step. This step tells you you're going through it, which is why it's so important that pyruvate gets regulated. Okay, you can enter the tricarboxylic acid cycle from acetyl CoA using fatty acids, so you can't regulate above that, or I should say you shouldn't regulate above that. You have to regulate below. So this is the regulation step. This step tells you we're going through, and we're gonna get as much NADH out of this as we can, so that we can oxidize that stuff later. In order to do that, we have to have oxygen. So pyruvate not being able, or pyruvate undergoing gluconeogenesis, or pyruvate going to lactate, or whatever the case may be, makes it great, because if pyruvate does go through with this reaction, we're going through it the entire way. Okay, and that can be difficult if you can't go through it the entire way. Okay, and it does mention some regulation. We're going to be talking a lot more about regulation in the future. Okay, so I want to point out they mentioned the Pro-R and the Pro-S arms. Those are not very commonly asked about. The reason I don't really like bringing them up is because they're just there kind of to distinguish between the carbons. They're not the most important thing. So yeah, okay. So I told you all that a conatase, the name of a conatase I think is absolutely fascinating. I've wondered this for a long time. Why isn't it just citrate isomerase? And the reason is because what's actually happening behind the scenes is two different reactions. The first reaction converts citrate to aconate. So this is the molecule, aconatase is the enzyme. Then the second converts that aconate into isocitrate. Okay, so it's a successive a dehydration and rehydration to isomerize for the overall isomerization. Okay, and this is going to be needed for future oxidations, which we're going to get to in the next step. Okay. All right. So I'm going to pause for just one moment, make sure that that's clear. Any questions, you're more than welcome to ask them. Okay, I'm assuming there are no questions. Let's move on. So aconatase is the first time we start talking about these iron sulfur clusters. And I'm going to talk about them a little bit, but they come up a lot next week, which is why I just want to introduce them briefly. Okay, so the first thing to point out is they're bound to the protein by cysteine residues. Okay, so cysteine has sulfurs. Sulfurs are great at heavy metal binding. There are 3 of them, and so they bind to 3 of the ions. Okay, these iron sulfur clusters, excuse me, are very, very good at providing charge. Okay, iron is very positive. Sulfur can go negative if it needs to be, but generally we're talking about the positive. Okay, the iron in the 4th position coordinates the groups, and so this is a great point for regulation as well. Okay, so we're going to talk about regulation later, but the point here is iron can be gained or lost, and the cysteine residues are the ones that are involved. The cysteine residues are the ones that bind these metal clusters to the actual enzyme. Okay? All right. So what does that have to do with citrate? This is the actual mechanism for what's happening. It's kind of complicated, but effectively all that we're seeing here is this iron binds to a single oxygen, and we draw binding, but in reality it's more of an ionic interaction. I'm just going to say binding because I'm definitely not going to clarify that every time because I'm going to forget at some point. But this iron is effectively binding to an oxygen, and in doing so, actually to 2 oxygens I should say, and in doing so stabilizes the leaving of one of them, which then allows a hydrogen transfer catalyzed by a base. Okay? That hydrogen transfer then allows a double bond to form here, which makes aconate, not the iconate. And so yeah, so this is kind of unrelated, or this is somewhat related to this reaction. So we've been talking about acetyl CoA. Acetyl and acetate are the same thing. They're effectively just a 2-carbon structure with a carboxylic acids. Acetyl CoA. So the only difference here is whereas this acetyl CoA has a sulfur and a CoA group, this has a fluorine, and that's it. So fluoroacetate makes an excellent inhibitor And as you guys can see, it's incredibly toxic by inhibiting the TCA cycle. And the reason it does that is because it's called it's a suicide substrate, which means that it basically binds almost completely as fluorocitrate. Okay. All right. So now we've gotten to isocitrate. Isocitrate is the reaction of isocitrate to alpha-ketoglutarate, or as it's also called, to oxaloacetate. I've never heard it called that in real life though, so don't worry about it. So this is the first decarboxylation that we're talking about kind of in more depth. We talked about very briefly pyruvate, but this is the first one of the actual TCA cycle. Okay, so two-step reaction, right? The first step, you make your NADH. And so when you make your NADH, you get, or I should say, you form this keto acid, okay? And because you have this keto acid, which is incredibly polar and drawing charge away, this incredibly polar carboxylic acid has less charge to draw away. And so by removing a hydrogen, it can be really easy to kick this thing off as CO2 and leave this carbon with, by introducing a hydrogen. I didn't mean to kick off hydrogen. By introducing hydrogen, because then this gains hydrogen to stabilize its electrons and we're happy, okay? The next reaction is very similar. To be fair, it's more similar to the Pyruvate Dehydrogenase complex, but the actual, in terms of what's happening, is very similar to either of the reactions that we talked about in terms of dehydrogenation. NAD plus goes to NADH. You end up adding a CoA group and removing a CO2. So whereas before we used a hydrogen to stabilize the carbon that lost its carboxylic acid, this time we use the S-CoA. And why do we do that? So let's think back. Yeah, we could use hydrogen or oxygen, but S-CoA is an activator, right? By attaching it to this carbon, we are activating this carbon in this molecule, and so it can do further reactions later. We're making it more reactive intentionally, okay? That's what we see here. So the free energy released by the hydrolysis of this can be used to drive GDP to GTP, okay? It's a very, very, very high energy substrate level phosphorylation. It's the only one in the TCA cycle. And then, like we said here, blah, 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 it makes ATP, okay? In plants and bacteria, oddly enough, you go straight from ADP to ATP. You don't have to use GTP as an intermediate, okay? Now, this is something I like to point out. I don't think it's ever going to be asked, but I think the structure for succinate is kind of beautiful because it's pretty much complete mirror image. It's a CH2 and then a carboxylic acid attached, and then those two are just attached to each other. It's very, very elegant. All right, so we've reached this point. Like we've talked about very briefly the synthesis of succinate, but we haven't talked about anything else. Let's pause and kind of give ourselves a little bit of an inventory of what we've done so far. Step one, we added 2 carbons to 4 carbons to make 6 carbons. We then got rid of the 2 carbons and we've gotten back to a 4-carbon unit. So in terms of carbon number, we've evened out. And the important thing with that is we no longer have those really high energy bonds to break. And so we're kind of back to neutral, right? We've made 2 molecules of NADH and 1 of GTP or ATP. And once again, that's per pyruvate. We can double that if we want the number per glucose molecule, okay? All right, so the next reaction is the only reaction, or I guess I should say not the only reaction, it's the only one in the tricarboxylic acid cycle. It's not the only one we've talked about today that involves FAD and FADH2. So very briefly, we talked about enzyme 3 of the pyruvate dehydrogenase complex that uses FAD to create FADH2. I briefly went over the alpha-ketoglutarate dehydrogenase reaction, and I just mentioned to you all that it's also going to have the same enzyme 3 as the pyruvate dehydrogenase. This is the only one that makes FADH2 as a kind of final product, is I think the most appropriate way to say this. And the reason is just seen below. The reaction is not sufficiently exergonic to reduce NAD+. Okay, so FADH2 gives us less energy, but this reaction doesn't make enough energy to give us this. Okay, all right. So Yes. Okay, so this is associated with the mitochondrial membrane, which will become really relevant next week. I'm never sure how much I should include about this, but this is one of the enzymes, one of the enzyme complexes in the membrane associated with the electron transport chain. Okay, and we're going to talk about the iron sulfur clusters for it next week, so don't worry about that. Okay, so we talked about flavo enzymes. I'm never sure how to pronounce that earlier. But effectively what they are is they use FAD. What is FAD? FAD is flavin adenine dinucleotide, okay? Now that encompasses something that includes AMP, okay? If you don't have AMP, you just have flavin mononucleotide, okay? If you don't have the phosphate, or should I say, if you don't have at least one phosphate, I should say, you have D-ribitol, and if you don't have anything, or if you just have the structure, you have isoaloxazine. None of this is very relevant, just the fact that you have flavoenzymes can be interesting, but ultimately, this is very rarely asked about. Okay? This has seen some questions. Okay? So flavoenzymes can take on 3 different states. You can have, so just very briefly, this is FADH2, this is FADH, okay? So the value of this is that you can have either a completely oxidized form, a completely reduced form, or what's called a semiquinone form, okay? This semiquinone has 1 hydrogen and 1 double bond, so it's a 1 electron reduction. It's the in-between, if you want to think of it like that. This can also this can also lose this hydrogen to become an anion. That's not the most important part, but really just the fact that it can either take on a single electron or a 2 electron redox reaction is really valuable for FADH2. Okay, and I say this specifically about FAD, but FMN is very similar. We just kind of lump them together because they're fairly similar for the purposes of this reaction, or for the purposes of this class, I should say. Okay. So this isn't the most relevant thing, but just the fact that there is histidine can be helpful to think about if you want to. I don't. Right, yes, and then iron sulfur clusters, like I mentioned, we're going to be talking about those next week. But the reason we're going to be talking about those next week is mentioned briefly here. The electrons from FADH2 enter the electron transport chain and contribute to ATP synthesis. This is really important, but it's also a topic for next week, so I'm not going to talk much about it. All right. Oop, too far. Okay. So this is, we didn't really talk about the last 2 steps of the reaction, but that's okay. So we're going to talk about for every acetyl CoA. So this is per 1 acetyl CoA, which is per 2 per glucose. But we're going to talk about glucose in this area right here anyways. Okay. So per 1 acetyl CoA, which is a 2 carbon unit, we get 2 CO2, so we completely use it up. We reduce 3 NAD plus 1 FAD, and we make the equivalent of 1 ATP. Okay. So now let's look at the net of glucose. So this is basically double this, add glycolysis, and then add pyruvate dehydrogenation. Pyruvate, and I'm just going to say dehydrogenation. Okay. So the reason I say we have to add all 3 of these is because let's think about this. We've got 3 NADH. We're going to double that to 6 because that's per 1. So per 1 glucose, so it would be 6. Then we get 2 from glycolysis, excuse me, and that's only 8. Okay. A lot of people, myself included at some points, have forgotten pyruvate dehydrogenase. Okay. That gives you 1 per pyruvate, which is the extra 2, which is why this number is 10 NADH. All right, so I told you all when we went through the Tca cycle, I walked you through 1, 2, and 3, the 3 CO2 groups that are removed. You have 2 pyruvate, each of them loses 3, you lose 6. So we completely oxidize glucose to get as much energy as we can out of it. Okay? We get a net of 2 FADH and 10 NADH. That's cool. And from glucose, we get 4 ATP. Okay? So the first 2.5, I'll call it, because glycolysis and Tca cycle, I'll call full cycles, and then pyruvate dehydrogenase, which I'm going to start abbreviating PD, is not a really full cycle. So from 2.5 cycles, we basically get 4 ATP. However, as you guys can see in this paragraph right here, which I wish I could just highlight all of it, but that might take me a minute. We're just going to underline it and call it a day. The NADH and FADH2 give us an additional 34 molecules of ATP. Okay, so 3 glycolysis TCA and pyruvate dehydrogenase combined give us 4, but the side products, the electron transport chain, gives us 34. That's huge. Okay, all right, so this is a tiny bit of that biochem has a bunch of connections everywhere picture that I'm sure you've seen. So I'm just going to very briefly mention pyruvate. We've talked about being really, really interconnected. This is just a little bit of how it's interconnected. It can form a ton of amino acids. Or I should say it's really connected to a bunch of amino acids because 3-phosphoglycerate connects serine, cysteine, and glycine, and it connects to pyruvate. Okay, and then obviously it also connects to phosphoenol pyruvate because that creates pyruvate. That's connected to the three aromatic amino acids. The TCA cycle, we've talked about oxaloacetate being important for phosphoenol pyruvate, but it's also important, as is fumarate, for aspartate, which can be used to make asparagine, or and from asparagine, the pyrimidine nucleotides, or any of these amino acids. Alpha-Ketoglutarate can help you make glutamate and proline, arginine and glutamine, along with all the purine nucleotides. So this is a huge section. And then Acetyl Coa connects us to fatty acids, to steroids. And I forgot to mention phosphoenol pyruvate and pyruvate connects to carbohydrates. So this is huge. And I'm not even mentioning porphyrins because we don't talk about those really at all. But this is huge. This cycle is really valuable for anabolism. It's really valuable for catabolism. It's very interconnected, and that's why it's such an important thing to talk about. Okay, all right. So when we use our intermediates, so I mentioned oxaloacetate is basically the starting point of the TCA cycle, or I should say the starting point of the loop of the TCA cycle, the circle, and that we just introduce acetyl CoA there for because it's convenient. You can make intermediates just in case you run out. So, and by intermediates, I really do refer to oxaloacetate in this case. So these are called anaplerotic reactions, and they help us. I don't want to use catch up, but they help us make up for anything that we're short. So we've talked about these reactions pretty much all before. The phosphoenol pyruvate to oxaloacetate we've talked about. We've talked about pyruvate to oxaloacetate. We've talked about malate to oxaloacetate. The only one left is pyruvate to malate. It's a very short reaction that just uses a malic enzyme. But yes, so CO2 provides us a carbon source, and we have some overlap with gluconeogenesis. We can also, I should say, use all of this backwards. So we've been talking about it going forwards, but you can also use it backwards if you're running out of malate or fumarate or any of the rest. Okay? All right. So anaplerotic reactions, I've seen a few questions about here and there. Usually just the name and a little bit of, this is the slide I was waiting for, but I wasn't sure where it was. So pyruvate, let me grab the highlighter. We use blue. I'm tired of yellow. Pyruvate carboxylase is the most important anaplerotic reaction. Great test question, right? It provides a direct link between glycolysis and the TCA cycle. I'm not sure why it just unhighlighted. It's very strange. There we go. And then keep in mind regulation. It's activated by acetyl CoA. So when you have too much acetyl CoA, you can just make more oxaloacetate. Very intuitive. It works very well. Okay. All right. So now let's talk regulation. So for the record, I know that I very, very quickly skimmed through these three and just kind of told you, here's what happens, let's move on. We don't talk about these reactions pretty much at all because they're fairly standard. And I apologize, I don't know if it's just on my end, but it seems to be erasing. As soon as I write something, I will kind of, okay. Let me make sure it's, okay. So this is a little bit unconventional, but I'm gonna apologize in advance. I'm gonna stop sharing this so that I can close it and reopen it. I know that that's not something I usually do, and it's not something I like to do, but I do want to make sure that you guys can see what I'm actually drawing, because that would be very frustrating if you couldn't. Okay, so let me reshare this. Okay, there we go. I do hope that it works, but if not, I will do my best to correct it. Okay. So I'm not going to go over the regulation too much. I'm going to go over a couple of the key details, and then we're going to move on, because I do realize that I'm going to run out of time if I don't. So very briefly, NADH is the goal of the cycle. So when you have high NADH, we stop. Okay, and it seems to be doing this again. So where are places that NADH regulates? NADH regulates pyruvate to acetyl CoA. Why? Because remember, pyruvate is the most connected metabolite. I keep saying that because it keeps being important. Pyruvate could be used to make a dozen things, so we shouldn't commit to making acetyl CoA if we don't need to. Okay, that's the logic there. NADH shuts down oxaloacetate to citrate, because remember, this was a very spontaneous reaction. We want to do our best not to make it happen if we don't need to make it happen. Okay, because it's not going to go. We can't go back if we do. Okay. The isocitrate to alpha-ketoglutarate, this is because this actually makes NADH, so that's why it makes NADH. Same with the alpha-ketoglutarate dehydrogenase reaction. It also makes NADH. Okay, I apologize. I'm not sure what's happening on my end, so this is rather frustrating. I'm going to do my best not to draw too much, but yeah, I apologize. Okay, and then on the flip side, NAD plus activates the cycle because it's telling you we need more NAD, or we need to convert some of this NAD plus to NADH. Let's do it. Okay. I mean, it pretty much focuses on the beginning steps because, once again, those are the steps that are going to kind of get us going. Okay. A couple of the really important ones to talk about are ATP and Succinyl Coa. If I'm not mistaken, yeah. So ATP inhibits the steps that provide either NADH or so that's pyruvate dehydrogenase and isocitrate dehydrogenase, or citrate synthase, which once again, very spontaneous, very, very, very irreversible. We want to make sure to only do this when we need to. Okay. Then finally, or not finally, sorry, AMP and ADP, they signal low energy status. Let's make more, right? All right. Finally, Succinyl Coa. Oh, my apologies. Okay. Succinyl Coa. Here we go. So that's a signal that the cycle is saturated. To be more specific, or I should say the reason that it's a signal that this cycle is saturated is because of CoA. So I don't know if you guys saw the, okay, well, let's see if maybe this drawing will stick around. CoA kind of goes in a loop like this for Succinyl Coa. If we don't have enough CoA to make more Succinyl Coa, we're going to get a buildup, or we're going to get a buildup of alpha-ketoglutarate, and we're going to get a buildup of Succinyl Coa. Okay, because we can't, or I should say, if we cannot make enough succinate, sorry, I apologize, I need to reword. If it's just so saturated we don't have enough enzyme to make more succinate, we're going to clog, we're going to have a buildup here. This buildup means we're not going to be able to make more Succinyl Coa because we're going to run out of CoA. So this is a great place to regulate because of that. Okay. So PDH is pyruvate dehydrogenase, and that can also be inhibited by acetyl CoA and NADH, which makes sense. And it can also affect phosphorylation. These are not the most tested I've ever seen. This is a great figure to kind of sum it up if you wanted to see it. Basically, if you have high NADH to NAD+, acetyl CoA to CoA thiol. So basically, if you have high NADH and acetyl CoA, so if you have high TCA reactants or products, you're going to shut this down. You're going to inactivate pyruvate dehydrogenase. If you have low NADH, low acetyl CoA, and high NAD plus or coenzyme A thiol, telling you that you need to activate the cycle and get stuff going. Okay? All right. Now, this is an interesting thing, and this is part of why I wanted to make sure I had enough time. The Glyoxylate cycle, and I'm going to mispronounce it a few times, is a modification of the Tca cycle that's used in bacteria, plants, and algae. Okay? And the reason that it is because they use their carbon they use their carbon elsewhere. Okay? They use their carbon for biosynthetic reactions in a way that we don't. And so the glyoxylate cycle is a way to help promote their acetate-based growth. Okay, so this is the glyoxylate cycle. As you guys can notice, there's some very similar aspects. So first, we start with acetyl CoA and oxaloacetate, and I'm focusing, I guess, on this area first. Okay, we start with the same two, and we go to citrate. Then we go to isocitrate. This is all the same. So this we are good on, and I'll even include malate here. These are the exact same. Okay, the place where it differs, and I really dislike that it's doing that. The place where it differs is isocitrate. From here, from here, so just so you guys know, this way is glycolysis. They're just showing that as a comparison. They form glyoxylate, hence the name, the glyoxylate cycle. From glyoxylate, they then introduce another acetyl CoA to help make malate. Okay, and then from malate to oxaloacetate. So let's look at carbon numbers. 4 carbons, 2 carbons, 6 carbons, 6 carbons, 2 carbons. The reason that it's 2 carbons, oh, I apologize, I misspoke. This is not a comparison to the, excuse me, this is not a comparison to the Tca cycle. This is, what is it called? This is a, I believe it's a hydrolysis, but it's a lyase reaction, so it's cleaving. And so what it's doing is it's cleaving it into 6 carbons and 4 carbons. So whereas we did that by, let's see if I can fix this mess. Okay. So we did that by just by releasing to a CO2. They do it by cleaving into glyoxylate. Why? To prevent carbon loss. Okay. Then, so fumarase is still 4 carbons. This is still 4 carbons. Okay. So what happens here is, if I'm not mistaken, we put in 2 acetyl CoA, CoA, and I apologize for the poor spelling. I'm not sure if it's gonna. Let me see if I can just fix that. Acetyl CoA, get out 2 malate. Okay. All right. So this is the actual cycle that you guys see here. Or we just get out 1 succinate and we can use that however we want to use that. I'm not 100% sure how the plants use it. Okay. So energetically, we make 2 NADH and 1 FADH2 per 2 acetyl CoA. In other words, per 1 glucose. Per 1 glucose, right? Because rather than using 1 acetyl CoA like we do, they use 2 acetyl CoA, and they only get this amount. As a comparison, we get 6 and 2 per the equivalent of 1 glucose. Okay, so we get, I believe, about 3 times as much energy from the Krebs cycle as they do from the glyoxylate cycle. However, they need their carbon for other things, and that's why they have to do that. Okay, so plants, glyoxylate cycle happens in the glyoxysomes. Makes sense. But depends on Tca cycle enzymes in the mitochondria. So that's a little bit of a weird thing, and I believe that's why plant structures are so different. Okay. Now I want to point out, glyoxylate can happen in the dark, and they use the acetyl CoA from fatty acid degradation. So this is the interesting thing here, is how things move back and forth between the glyoxysome and the mitochondria. So we're going to start with succinate, because I think succinate is the easiest place to start. Succinate travels from the glyoxysome into the mitochondria, and then we go up to oxaloacetate. Okay, so what this is saying is you take the glutamate from the glyoxysome and move it in, and then these two together give us aspartate and alpha-ketoglutarate. which can then transfer back into the glyoxysome as, well, the aspartate stays and the alpha-ketoglutarate stay, and they reform glutamate and oxaloacetate. So this entire process is a way to get oxaloacetate from the mitochondria into the glyoxysome, and then get the succinate out of the glyoxysome and into the mitochondria to make more oxaloacetate. Okay, so the process there is basically succinate into the mitochondria to make oxaloacetate, oxaloacetate and glutamate transferred and then remade, and glutamate is just transferring back and forth basically. It goes to the right into the mitochondria, and then the two together go back into the glyoxysome, and then you remake oxaloacetate and glutamate so that oxaloacetate can be used. Okay? All right. So PEP carboxykinase. This is a bit of an interesting fact. It is not the most important thing that you're going to be tested on, so not much to worry about. But basically, when it's overexpressed in skeletal muscle, let's think about why that is. No lactate generation. I mean, it's kind of the key here, right? It helps us to have fast reactions that can be done instead of lactate dehydrogenation. Generation, I guess. So this is because mice can do fat generation during exercise, can use fat degradation for energy during exercise, but we can't. Okay. And these are some pictures about that. I'm not really going to spend time on this. Oh, okay. This was me summing stuff up, and I totally forgot to erase this in my notes. I don't think some of these slides have loaded, actually. Okay, that's fine. So what I told you all we would do, because we have about 10 minutes left, is let's kind of do a bit of an inventory as to what our options are with glucose. Okay, so we're going to make our own web. Glucose here. Glucose could be stored as glycogen. It could stay in the blood, and I'm gonna say like this is glucose in the blood. Okay, could stay in the blood. Maintain our blood sugar. When it gets too high, let's store it as glycogen or let's use it. It can go to the muscles. Oh, and this is liver. It can go to the muscles to give us glycolysis, right, into pyruvate. Pyruvate could either go back to glucose through gluconeogenesis if needed. Neogenesis, right? Pyruvate can then either go to lactate, still in the muscle, in the cytoplasm of the muscles, or, so this is no oxygen. This is with oxygen. Pyruvate can go through the Tca cycle and electron transport chain. Okay, what else can glucose do? Glucose can go into the pentose phosphate pathway to give us R5P and NADPH. That is horribly misspelled. Just gonna try that again. NADPH. Okay. All right. Now let's talk energy. Okay, we're not going to focus on PPP or glycogen because we've talked about those, and I believe those were your last exam anyways. But let's do a quick recap of everything we know. Glycolysis. We start with a 6-carbon unit, and we make 2 3-carbon units plus, a net of 2 ATP. So we use ATP and we make ATP. So I'm gonna stick to the net of 2 because we make 4, we lose 2. Hope that's okay with everyone. And we make 2 NADH. 2 NADH. Okay. So that's kind of the inventory for glycolysis. Next, pyruvate dehydrogenation. We use 1 3-carbon unit. And we make CO2, Acetyl Coa, which is a 2-carbon unit, and NADH. So I'm going to go ahead and double this and say 2 of these, 2 of these, 2 of these, and 2 of these. TCA gives us per 2 carbon acetyl CoAs, gives us 6 NADH, 2 FADH2, and 4 CO2. I knew I was forgetting something. Okay. And then from there, or I guess I should say, what is the net of this? So let's start off by just kind of combining terms, and I'm going to highlight stuff to make things easier. 6 carbon, and then I'm just going to highlight anything that has to do with carbons. 3 carbons, 3 carbons, and there's 2 of them and 2 of them, so these guys are going to cancel actually. Oh, oops, I meant to switch to a marker. So 2 carbon and 2 carbon, those are going to cancel. All right, 2 carbon units. We have 2 carbon units here, so they'll cancel as well. So effectively what we have is, from all the carbon stuff, we have 2 CO2 and 4 CO2. And I know that the figure did include this, but I just want to make sure we go over it in depth, because this is a question that I think a lot of people have. So if there's anything you're missing here, please ask. Okay. Oh, I also forgot the ATP, didn't I? 2 ATP. Okay. All right. So now we'll combine the terms. All right. So now we'll work on green actually. So that green will be ATP. There's ATP here. There's ATP here. Let's move on. Okay. Blue will be the NADH. There's 2 here. Oh, I did not switch to blue. There's 2 NADH here. There's 2 NADH here. There's 6 NADH here. All that's left is the FADH2, and we don't need to highlight that one, or actually I will just for completeness. Okay, so what we start with is glucose, which is the 6 carbon unit. And that's it, really. We do have like NAD plus, stuff like that. I'm kind of ignoring those. We get 10 NADH, 2 FADH2, 4 ATP, and 6 CO2. So when I mentioned at the very beginning, we oxidize glucose completely, this is what I mean. We start with 1 6-carbon chain. Every single one of those carbons gets as many oxygen bonds as it possibly can, which is CO2, which is 4 bonds to oxygen. So it's oxidized completely. Okay, and then obviously these two go on to make 34 more ATP, but we're not talking about that right now. Okay, so questions on the tricarboxylic acid cycle. That's something I wanted to dedicate a little bit of time to, because you're going to get some. So very briefly, a couple of questions that are totally fair game are just anything to do with the cycle. So why is what enzyme is this one? And it's a kinase. What enzyme is whatever, things like that. Which one is very, very spontaneous and can be irreversible? Oxaloacetate plus acetyl CoA going to citrate, right? Those are fair game. Which is the only substrate level phosphorylation in the TCA? Succinyl CoA to succinate. What's used as regulation? All of these things, totally fair game. But there's one more type of question that some of you who've had Bio 1 might be familiar with. If I start with 10 molecules of alpha-ketoglutarate, how much energy will I get at the end? Okay. Or I guess I should say, what will the products be? So however many molecules of alpha-ketoglutarate. And this is important for 2 reasons. The first is because we do sometimes do that. If we go down toward the bottom to, any second now, apologies. Nope, not this figure. Nice. Oh, I did skip these. I apologize. I skipped these and I didn't even notice. There's not really much here though, to be honest. It's okay. Okay, this. So we do sometimes start from an intermediate. So you're not going to be asked for the depths of, like, if I have proline and I want to convert it back to a Tca cycle intermediate, what would I convert it to? But you could be asked, I convert one of my whatevers, one of these things, and they don't have to list it, but they could, back into alpha-ketoglutarate to perform, to go through the Tca cycle. How much energy will I get out of it? Or maybe they'll ask, I converted something over here back into fumarate, excuse me. How much energy will I get out? Or something like that, right? All of these questions are fair game. And I wouldn't expect too many of this type of question because it takes some math. But the big things that I would focus on are enzymes, the kind of notable reactions for any reason. So Succinyl Coa to succinate, Oxaloacetate to citrate, Isocitrate and Alpha-Ketoglutarate dehydrogenations. Those four that I would focus on especially. Anything to do with regulation, I would know forwards, backwards, upside down, etc. I would understand Pyruvate Dehydrogenase, understand that enzyme in the complex well, or I should say those enzymes in that complex. And then what we're going to pick up next week, so don't worry about this part too much, but what we're going to pick up is the iron-sulfur clusters, the whole transfer of electrons, and then one of the most important topics is succinate dehydrogenase. I'm sorry, not succinate dehydrogenase. Excuse me. No, that's correct. Yes, succinate dehydrogenase. I apologize. I always blank out on those two enzymes. This enzyme right here. And we're going to talk about this one next week anyways, the whole associated with mitochondrial membrane. But those are some of the keys that we're going to be focusing on. Next week, I'm going to admit, is one of my least favorite weeks. It is hard. The content is not easy. It's not very intuitive. But you know, come to the review or watch the review on YouTube, and we'll do our best to help you out. Okay. And I see that I'm starting to run out of time, so I will pause for a moment for questions. But if there are none, then thank you all for joining or watching this on YouTube if you are. If you have any questions, please feel free to come to the tutoring center. We're open 11 to 8 Monday through Thursday, or 11 to 5 on Fridays and Saturdays. Thank you all and have a great rest of your day. Bye.